All right, so welcome to week 4 of microbiology lecture. Today I will be covering one chapter for this week. This will be chapter 13, which is all about the microbe-host interactions. And the reason why I'm only covering one chapter is because you guys have an exam, your first exam this week on chapters 1, 3, 4, 6, and 7. So getting right into it, so microbe-host interactions. So I'll be covering today all of the steps that a pathogen will take to cause disease. So this involves the following steps. The microbe making contact with the human or the animal host. So how does it enter? What is the portal of entry? How does it then get to its target cell or target tissue and start to invade that particular area of the host? So at first we'll colonize with the native flora or the normal flora, and then how does the pathogen cross the lines of defense and enter sterile tissues or tissues where it will actually indeed cause infection? Now the state of infection is referring to that condition where the pathogen is replicating within the tissues. It is eliciting its disease-causing effects by the presence of particular virulence factors that I'm going to talk about. It may also release toxins, which can lead to intoxication and can cause inflammatory responses and cell or tissue damage. And then how does the native immune system respond? So there are beneficial effects here where the immune system could repair the damage, or we could have a state where the effects of the microbes result in injury, disruption to tissues, and potential mortality. So I'm going to talk about all the details with regards to every one of these steps along the way. Now, remember, since we're talking about today how pathogens, disease-causing microbes, can actually enter a human or an animal host and make that host sick, remember that we have 10 to 1 good microbes or normal commensal microbiota as compared to our own animal eukaryotic cells. So all of the majority are bacteria, fungi, protozoa, viruses. All of these are residents, they're normal, and they carry out this process called microbial antagonism. So this is just a fancy way to say that the good flora, the good bacteria, mostly fungi, protozoa, and viruses, benefit us by preventing the overgrowth of the harmful ones or preventing the overgrowth of the pathogens. And remember, they do this through a variety of mechanisms. So they are able to produce the majority of IgA antibodies or immunoglobulin A antibodies, which provide mucosal immunity. They also can produce their own antibiotics. They elicit these protective immune functions, especially within the gut environment. So they are critical for maintaining our health. Now, when do we actually start to become colonized with the normal, healthy microbiota? It's actually just before birth. So the uterus, as the fetus is developing in the womb, and the contents are normally sterile, but the breaking of the fetal membrane will expose the infant. So as the infant passes through the birth canal, that's when the colonization of the normal microbes begins. And all subsequent handling and feeding continue to introduce what will eventually populate the body in the form of the normal commensal microbiome. Now, it's actually really interesting if you guys even Google this. If you look up, there are a lot of scientific articles coming out recently suggesting that children who are birthed via C-sections, so they circumvent passaging through the birth canal, have differential population of normal microbes as those who are born via normal childbirth through the birth canal, and that they're finding that it may be a potential detriment to infants that are born via C-sections because their immune system may be lacking from the different microbes that are colonized early in life. And there is some scientific evidence for that. Now, as far as the sites of the body that are colonized by microbes, I spent a good amount of lecture one kind of stressing this idea to you guys, but just as a review, I mentioned all over the surface of the skin, so staph for sure, Micrococcus, Corynebacterium, Propionibacterium, strep bacteria, even Candida and arthropods, Dermodix mites, these are present in sebaceous glands and also hair follicles. So these are normal. Within the GI tract, especially the large intestine or the colon has the majority of bacteria and very diverse. Strep, Neisseria, Fuso-lactobacterioids. These are also found in the mouth actually. And I should mention Bifidobacterium, Clostridium as well. Escherichia is another one that you would find there. And then also in the throat, so the upper respiratory tract. Now, we don't see lower, but I have seen a paper recently, an article that came out citing normal microbes that live within the lungs, although these populations are much lower, but there is evidence for that. Also, genital tract, especially the lacto and the strep bacteria, even the urinary tract, staph, strep, lacto, again, and also the eyes and ears. So for the eye, staph, strep, Neisseria, and actually the eyelids and the follicles have similar microbes as the skin, but the deeper tissues are found to be sterile. And then also the ear. You find even fungi here, Aspergillus, Penicillium, Candida, some other yeasts as well. So remember, all of these anatomical sites are primary sites here for the normal microbiota, the commensal bugs that actually are kind of serving as the first line of defense to disease. Now, as far as the bugs that can actually make us sick, there's a differentiation here between pathogens that will make a healthy person sick versus those that will specifically cause disease in someone who has an immunocompromised status. So, a pathogen that will make a healthy person sick is referred to as a true pathogen. So this is capable of causing disease in healthy people with normal immune defenses. And some examples of this, if you're not immunized or vaccinated, the flu, plague, malaria. But this is different than an opportunistic pathogen. So if you are severely immunocompromised and we see, for example, late-stage HIV patients when they're in that third tertiary clinical full-blown AIDS stage where they have basically next to none as far as their CD4 T helper cell. So their white blood cell count is almost completely wiped out. They are just riddled with all of these different opportunistic infections. And in the case of an opportunistic pathogen, these can cause disease when the host defenses are compromised or when they're growing in a specific area of the body that is not natural to them. So some examples of this, Candida albicans, which causes a yeast infection. Pseudomonas is another one that can cause a multitude of skin infections. Whether you are a true pathogen or an opportunistic pathogen, the severity of the disease state which you would cause upon a host would depend on the virulence factors that you possess. And another word for severity of disease or the potential magnitude of disease is virulence. So that's analogous with severity of disease or the pathogenesis. So if you hear me say that this specific factor increases pathogenesis or increases virulence, that is just referring to the severity of the disease. And I'm going to talk about different types of virulence factors that microbes possess. All right, so I mentioned a couple slides ago, this whole chapter is all about how a microbe makes you sick, right? Well, if it's going to cause disease within an animal or a human host, it needs to enter your body first. So there are multiple portals of entry. And this is just referring to the route with which a microbe will take to enter the tissues of the body. So this would include, you can see here on the figure on the right, but they're listed here, through the skin. So this could be through a cut, a nick, abrasion, puncture, incision. This also could be from food or drink, right, through the GI tract, or inhaling something through the respiratory tract, oral or nasal cavity. Also through the urogenital tract, so STDs or displaced organisms in the case of urinary tract infection, or transplacentally as well. Now, a couple terms here that you may see throughout the lecture: exogenous versus endogenous. If it is an exogenous agent, this originates from outside of the body, versus endogenous, which would be already existing on or within the human body. Now, every pathogen has what we call an ID. So an ID is referring to the infectious dose. And the infectious dose is the minimum number of microbes required to establish infection. So basically, if you are exposed to, if your body comes into contact with a number that's lower than the ID for a pathogen, you will not get sick. Now, what I would like you guys to remember about infectious dose is that microbes that have a very small ID, meaning you only have to be exposed to, let's say, 1 or 2 of that pathogen, these guys usually have a much greater, more severe disease course. They have greater virulence. And again, if you're not exposed to that number, that minimum amount of the dose of that pathogen, you will not get sick. Now, this is just a table here showing you kind of a survey of different IDs. So if you look at measles, it only takes 1 virus. This is associated with higher virulence, right? Much higher virulence than, let's say, typhoid, which is 10,000 cells, right? Q fever, 1 to 10 cells. That is also associated with higher virulence. So you see cholera takes a whopping 100 million cells, and you actually have to ingest these, and it's usually through contaminated water. Now, once the pathogen actually enters the host, it will attach to a specific area of the body. So it will do this via this process that we call adhesion or attachment. Now, adhesion usually is made possible via the potential for any one of these microbial structures. So they're all different depending upon the guys at the bottom here. Usually we see in helminths or parasitic worms. Cilia, remember, we don't see cilia in prokaryotes. We see cilia in eukaryotic cells. But all of these I mentioned in the lecture where we focused on prokaryotic structures. So bacteria have fimbriae. They have these proteinaceous components that they can use to attach onto a specific surface. So E. coli, for example. E. coli accounts for a large majority of at least 50% of urinary tract infections. And that particular strain that causes a urinary tract infection usually will adhere to the bladder wall via these fimbriae. Also, if it's causing enterotoxinogenic or an enteroinvasive, a GI traveler's diarrhea case, the fimbriae also will work to attach to the inner lining of the intestinal wall. Also, flagella can be used for a mode of adhesion, or those slime layers or capsule layers. Remember, these make up that outer layer, the glycocalyx, which is a sticky layer that can be used for attachment. So during this specific stage of adhesion, the microbes are gaining a stable foothold at the portal of entry. And this is dependent upon any one of these specific molecules and how they're interacting with the host. Now, here's some examples of what I just cited here. The fimbriae attaching to, let's say, an intestinal epithelial cell wall, or the capsule attaching to the same surface. Or in the case of a virus, we can add spikes to that list. So the glycoprotein spikes that you see on the surface of the protein capsid of any virus can be used to bind to unique receptors on the host cell to actually gain entry into that host cell. Now, when the pathogen enters the host through a portal of entry and attaches to the host cells, then you have the initial response of the immune system of the host defenses from white blood cells called phagocytes. Now, because this process is a native, natural immune response, there are some clever pathogens that have evolved the ability to produce what we call anti-phagocytic factors. So these are used to actually avoid the process that's elicited by these phagocytes. So certain strains of Staphylococcus, also Streptococcus, produce these particular compounds called leukocidins, which actually are toxic to white blood cells. They can kill white blood cells. Also, any pathogen that has a slime layer or a capsule makes phagocytosis very difficult. So what is phagocytosis? Phagocytosis is basically a way, if you see on the right-hand side here, it's a way by which the white blood cell can actually engulf a pathogen. So the mechanism by which phagocytosis is elicited is from the interaction between the pathogen and the phagocyte via an antibody bridge. So the immune system will produce an antibody that's going to target and bind to, on one side of the antibody, the pathogen. The other side of the Y-shaped antibody will bind to the phagocyte, and this creates a bridge. It creates a bridging mechanism to pull the pathogen into the phagocyte, because both of these cells are negatively charged, so they repel one another. And each portion of this antibody on this side and the Y side of the antibody is positively charged. So it's attracted to the negative charge of the pathogen here, the negative charge of the white blood cell. So it creates a bridge. Now, like I said, some pathogens are able to avoid this via slime layer or these anti-phagocytic factors. Or the other clever way that pathogens can circumvent this anti-phagocytic factor as being a negative response would be that some pathogens actually like to replicate within phagocytes. So they would rather replicate within the white blood cell and then cause the bursting or the lysis of that white blood cell. So it kind of tricks our cells into thinking that it's engulfing the cell and killing it when really it starts to replicate. Now, there are many different types of virulence factors. So remember, what is a virulence factor? This is something that is going to contribute to the severity of disease. So it's a trait used to invade and establish themselves in the host. Also determines the degree of tissue damage. Now, what are the different types of virulence factors? Okay, different types of virulence factors can be the following. Number one, enzymes. So this could be, for example, DNase or collagenase. These are enzymes that can actually break down tissue or can break down DNA, and it permits the invasion of pathogens. So number one, enzymes. Number two, toxins, endotoxins or exotoxins. These are different depending upon how they are released from the particular bacterial cell. So in the case of an endotoxin, it's actually released from the cell membrane, from the lipid A portion of gram-negative bacteria. Exo is secreted from the intracellular contents of the bacterial cell. So this would be either gram-positive or gram-negative. But lipid A endotoxins are specific to gram-negatives. So virulence factors would be 1, enzymes, 2, toxins, 3, antiphagocytic factors. So this would be the leukocidins, for example. Helps them to kill or avoid phagocytes and remains an irritant to host defenses. Now we also could add, in the case of virulence factors, the adhesion factors, right? The Fimbriae or the spikes, those would also be considered. So that would be a number 4 on the bottom of the list here. All right, so here are a few cartoon illustrations of an exoenzyme at the top. So these little green circles are the representative pathogenic bacteria that are releasing a toxin from the intracellular contents into the external environment, and this toxin will destroy the cells or the tissue in the surrounding environment. So for example, hyaluronidase, which can be produced from strep bacteria, or coagulase, which is produced from Staph aureus strains, can cause tissue damage in the surrounding environment. Now, this is similar to how an exotoxin is released and how it elicits its disease-causing effects. Now, the endotoxin is a little different because the endo, remember, is within the lipid portion of gram-negative bacterial cell membranes. And on the bottom, you see here this antiphagocytic factor, such as a capsule or a slime layer, which will actually block the process of the white blood cell trying to engulf that pathogen. Now, as I said before, some pathogens actually like to replicate within the white blood cell. So with regards to exo versus endotoxin, it takes more of a higher dose of the endotoxin to actually exert its toxicity effects. Usually the symptoms associated with endo are fever and an inflammatory response, whereas the exo is specific to the target cell type. And remember, endo is derived from the LPS of that cell envelope of gram-negative. They're more stable, and they cannot be converted to a toxoid, whereas the exo can. And also, in the case of the sources, remember, endo is strictly found in gram-negative, whereas exo can be both gram-negative and positive. Now, this is important here. I'm going to go through the 4 distinct stages of clinical infection. So this is after the pathogen has entered the human body, it has adhered to the target cell or tissue type, and we've had an immune system response. What are the 4 specific stages of the infection? And the 4 are the following. Number 1, the incubation period. So this is the time from when you are exposed to the pathogen until you see your first symptoms. Now, throughout this period here, the pathogen is multiplying, but it's not until we get to the period of invasion where we have high levels of the pathogens, and that's when we see fever, we see the rash, we see some of the signs and symptoms. And I think we've all had a cold or gotten sick at one point or another, and right before we've come down with that infection, we kind of have this sixth sense feeling that something is wrong. So this is what we call the prodromal stage, where it's a vague feeling of discomfort, no specific complaints. And then again, this progresses into the point where you're actually seeing signs and symptoms of disease, and hopefully followed by the recovery or convalescent period. So this is what it would look like if we graphed it here on the y-axis, the intensity of the symptoms, and also we can correlate this with the number of the pathogen, the amount of the pathogen that is present over time. So if we have that initial exposure to the microbe, so this would be the portal of entry here, it enters the human body, then it's starting to replicate, but it's at a very slow rate. It's the incubation period. So we actually have contracted that bug or that pathogen, but we don't have any signs or symptoms. And then after a certain period of time, now these durations are different for every pathogen. So the incubation period for a specific viral infection could be very different from a specific bacterial infection. They are all unique in that sense. After we cross over the incubation period, still no symptoms, we start to have this general feeling of discomfort. So the pathogen is slowly starting to replicate at a higher, gradual, more progressive rate. And then similar to the log or exponential phase of bacterial growth, it's replicating at an exponential rate. And we start to see the signs and symptoms in this pink window here until we reach this height of infection, where then hopefully the immune system has been kicked on enough to start to combat the infection, and hopefully the amount of the pathogen goes down at an exponential rate over time and we recover. So this would be the convalescence. Now, there are many different types of infections that we could experience. So we could have a localized infection where a pathogen enters the body and remains confined to one tissue. So this could be an infection of the skin, for example, or even a respiratory infection. This is different from when the pathogen is spreading throughout the body. So it could spread to several sites, and the scariest is when it will spread to the bloodstream, which we can then refer to this as a patient or a host going septic. So when it's in the blood, if it's bacterial, that would be bacteremia. If it's a virus, that would be viremia. And at that point, when a patient is septic, that could lead to organ failure, shock, and even death if it's not treated right away. Now, focal infection is referring to an infectious agent that breaks loose and is carried elsewhere. Mixed infection, an example of this, it's also called polymicrobial, would be, for example, PID or pelvic inflammatory disease. So this is a disease characterized by a co-infection with two different STD causing agents such as gonorrhea and chlamydia. Usually women who are co-infected with both of those end up with fallopian tube issues, scarring, infertility. So usually polymicrobic or polymicrobial infections can cause more lasting scarring or damage. And then also we have primary versus secondary. This is just with regards to over time, you have an initial infection, and then as your immune system is weakened, you may have a secondary infection. So at this point, if you have two microbes, then it would be polymicrobial. And then also with regards to severity, you could have an acute infection, which comes on rapidly and may be severe, but it does not last very long. So temporary versus chronic, which persists over a long period of time. So hepatitis, for example, Hep C, there's no cure, there's no vaccine. People who are infected have symptoms, they progress over a long period of time, and they can end up with liver damage, liver cancer, liver nodules, or hardening of the liver over time. So these are all the pictorials here of it on the bottom. So this localized, one site, systemic, throughout the blood, focal, it starts off in one site and spreads to a completely different area. So this could be even from, for an example, in the case of dental work, you could have an infection in the mouth or a microbe, a pathogen in the mouth that through a cut in the gums, especially for people who have gingivitis or periodontal issues, could enter the bloodstream and spread to the heart, causing endocarditis. That could potentially be a severe life-threatening condition. Or mixed infection, you could have multiple microbes causing disease. And then primary, UTI, for example, usually people are prescribed antibiotics that then cause changes in the pH in the vagina, and then a yeast infection could result. So that would be a secondary infection. Now, big difference here. So during the period of invasion, there's a big difference between the signs and symptoms that are recorded or observed by a particular patient. So if we are in that period of invasion where the pathogen is replicating at an exponential rate, an individual will perceive a symptom. This is subjective. So they will report that they have particular symptoms such as chills or a headache or fatigue, but these are all subjective. There's really no way to measure these. Symptoms are different than signs. So a sign would be something that is actually observed by the patient or a healthcare provider. It's objective, right? So you're seeing that you have a high temperature. You're seeing that you have a rash, or the doctor or the pathologist will look in the blood, check for the presence of antibodies, or check for the presence of white blood cell count, or also even see the presence of the bacteria in the blood, or hear in the chest, right? Perturbation of breathing or even heart rate. So these are all signs. Now, the earliest symptoms of disease as a result of the immune system being activated, usually fever, also pain, soreness, swelling, and signs of inflammation could include a medical term, edema, which is accumulation of fluid, also abscesses. So these are walled off collections of inflammatory cells, and also the pathogen itself, and also swollen lymph glands or lymph nodes is another characteristic sign of an infection. Now, sign, remember, this is objective. This is something you measure. You may also see as evidence to support a sign of sepsis or infection in the blood. Look at the number of white blood cells. So an increase in these would be leukocytosis, a decrease leukopenia, and that term sepsis or septicemia is referring to a microbe in the blood. If it's a bacterium, bacteremia. If it's a virus, viremia. Some infections may go unnoticed because they are asymptomatic. So you may be infected with a particular pathogen and not know it. There are a lot of viral infections that behave that way, and even bacterial if you end up becoming a chronic carrier. It could be a very brief period of illness and you don't realize that you are sick, and then you may be passing on this infection to others. So if you don't have the symptoms, right, people don't seek treatment, they don't get tested for a large majority of them. So I covered the aspects of the different phases of the period of invasion, right? So we said that it's incubation, prodromal, and then you have the period of infection or invasion followed by convalescence or recovery. Now, hopefully, if the individual recovers, the immune system responds. The body will find a mechanism to rid itself of that pathogen through a portal of exit. So this is the way in which a pathogen will depart from the host, and this will greatly influence how the pathogen is passed to others. So, for example, it could be through the respiratory system, coughing or sneezing, through mucus, sputum, through the nose, nasal drainage, or saliva. Also, skin scales. Skin cells and open lesions. Fecal exit, so there are a lot of oral fecal routes of a particular, especially with foodborne illness, modes of transmission. Also through the urogenital tract for especially sexually transmitted diseases, and also blood transfusions, removal of blood is another mechanism. And also insect bite, so an insect could bite a host and actually contract that particular pathogen and pass it on to its next host that it will feed on. Now, some pathogens will behave in a latent manner. This involves lying hidden, basically, within the host. And this can result in future outbreaks. So, for example, herpes, HSV1, HSV2, which are the causative agents of oral and genital herpes, respectively. For patients who are infected with those viruses, they may have an initial outbreak, so there's initial symptoms, and then it can be later reactivated in the form of an outbreak over time. So it's a stimulant or a trigger for some of those outbreaks are could be stress, could be in women the menstrual cycle, or exposure to UV light. And during those active stages, the pathogen is shed, but in some cases it could be shed even when it is not active. So some people think that, they're not contagious during those stages, when in reality there is a low amount of shedding that's at least above that infectious dose number that we talked about earlier. Now, another example of a latent type of a pathogen would be the virus that causes both chickenpox and shingles. So varicella zoster causes chickenpox, usually at a younger age, and then it lies latent and then is later reactivated and lies latent in a nerve system, later to be reactivated as shingles. Now, some people can also serve as chronic carriers that are shedding the agent, and this would be especially for a lot of bacterial cases of this, the classical one, which you guys will be watching a video on Typhoid Mary. She was a chronic carrier of the causative agent of typhoid fever. So she was shedding Salmonella typhi in her feces, and she was actually spreading the bacteria when she was preparing food to people who were ingesting that bacteria, and then they were coming down with the disease. So she was a chronic carrier. She didn't actually display the symptoms herself, but there are cases of this. And then especially in these long-term infections, especially the latent infections or progressive infections, can lead to permanent damage to tissues and organ systems. Now, it's important to note here, every pathogen has a reservoir, and the reservoir is a primary habitat in the natural world. So this is really important because when you have pathogens that have non-human reservoirs, especially other animals, and if it's more than one animal, or if it's a non-living reservoir like soil, water, this becomes really tricky to eradicate or completely get rid of a particular infectious disease, because if the only reservoir is in a human, you can vaccinate everyone and you can eradicate that disease. But when you start talking about having a pathogen living in animals other than humans, you really can never get rid of that pathogen completely unless you get rid of the source of the primary habitat. So it makes it a little more tricky. And then this term source is an individual or object from which an infection is actually acquired. So here's the little chain of infection here from a reservoir, right, which could be a human or an animal or even a non-living host here, host reservoir. And then we have the mode of transmission through direct contact, a vector, which would be an insect, food, airborne droplet to a susceptible host, and then it enters through the portal of entry. And then we cycle through all those steps that we just talked about, the 4 stages of disease, right? So in a living reservoir, there are a few differences here with regards to an asymptomatic carrier can actually spread potentially the infectious agent during the incubation period, or even in the recovery period. So that misnomer, that misconception about, oh, I'm on antibiotics, I'm getting better, I can't spread this disease, may not always be the case. Sometimes individuals can still spread the pathogen or transmit the pathogen even when they are in that recovery period where they don't have symptoms. Now also in the chronic carrier, like the case of Typhoid Mary, she was shedding the pathogen over a long period of time and not knowing that she was infected herself. And this is a big one on the bottom here, passive carrier. So for anyone going into nursing, into the healthcare field, this is a huge problem. So I'm not sure if anyone saw a recent news on the news and the recent finding citing the fact that Connecticut is the worst state in the country for hospital-acquired infections. So the numbers are astronomically high for people who are contracting infectious disease from healthcare workers, from patient to patient in a hospital environment. So this is something that we really need to work on to try and get those numbers down. If it is a hospital-acquired infection, it's called a nosocomial infection. So this is a term that you guys should remember. Now, this is just a cartoon diagram showing you the different types of carriers here, right? So asymptomatic, and the star is representative of the pathogen. During incubation, right, you don't know that you're infected, or during recovery, or a chronic carrier, usually through food preparation, passive carrier here in the case of a hospital-acquired infection. Now, in the case where an animal is the source of the natural habitat or the reservoir for a pathogen, if it's a live animal other than a human, it's called a vector. Now, the majority of vectors are insects. So these are fleas, mosquitoes, flies, ticks. There are at least a few diseases in Connecticut that pretty high incidence here in the state that are vector-borne diseases, like Lyme's disease. I know that's been on the rise. Also, in the case of mosquitoes, West Nile is a newly emerging infectious disease. And also, some larger animals can spread infection. So other mammals, birds, lower vertebrates as well. And they're all different depending upon the pathogen that we're talking about. So when we get to the actual chapters where we're zooming in on each pathogen and the disease that it causes, I will cite the particular vectors for each disease. Now, if it's a biological vector versus a mechanical vector, very different with regards to the pathogen's life cycle. So for example, in the case of malaria, malaria is transmitted via a biological vector, the mosquito. The mosquito will actually contract that particular pathogen. It's a protozoan. It's called Plasmodium vivax. And Plasmodium vivax will replicate within the mosquito's gut. Once the mosquito actually transmits that pathogen to humans, there's another biological cycle of the parasite, of the Plasmodium vivax, that occurs in humans. But the point here is that the pathogen is actually replicating and growing within the mosquito. This is different than a mechanical vector. In the case of a mechanical vector, this would be at a summer barbecue. You have a fly that lands on a pile of poop in the yard and gets some bacteria on its legs and then flies over to your potato salad and transmits that bacteria on its legs. But the pathogen is not actively replicating within that vector. So it's not necessary to the life cycle, and it's just transporting it without being infected. We also have a type of an infection called a zoonotic disease, or a zoonosis. And this is an infection that is native to animals but is naturally transmissible to humans. So an example of this would be Lyme's disease or dengue fever. And these types of diseases are not transmissible from human to human. So humans don't transmit the disease to others, but they contract them from animals, and usually it is through an insect vector. Now, there's at least 150 zoonotic diseases worldwide, and they're definitely on the rise. They make up 70% of all new emerging diseases worldwide. And this is going back to what I said about when you have a reservoir other than a human, it's impossible to get rid of the disease without getting rid of the animals that are actually harboring that particular pathogen. There's also an example where you could have non-living reservoirs. So sometimes the natural habitat for pathogens exists in a soil environment, or even water, or even in the air. All right, so if there is a pathogen that is communicable, that means that an infected host could transmit the agent to another host. And when you have one that's highly communicable, that means it's contagious. There are some diseases that are non-communicable, right? So we just talked about the zoonotic. It's non-communicable from human to human. Once one human is actually, a single host is infected, they cannot infect another host. And this usually occurs when a person is compromised, and let's say they have an opportunistic infection, or if it's with an organism in a natural non-living reservoir as well. This may be the case. So how do you contract a disease? What are the potential patterns of transmission? It could be through direct contact. So this could be through physical contact or fine aerosol, also indirect. So this could be through fomites, inanimate objects, also food, water, biological products, or in the air. So droplet, nuclei, or aerosol. So these are all the types of transmission routes, and these would both fall into communicable diseases, right? So direct would be through kissing, sex, droplets like colds or chickenpox, HIV or syphilis from mother to fetus, and then also biological vector like a mosquito, which could transmit West Nile virus. Or it could be indirect through fomites like a doorknob, inanimate objects. This would be a staph infection, or fecal oral contamination. This would be through food handling, which can cause many different types of food poisoning cases, and also in the air, TB, the flu, hantavirus, even out in the Midwest, where usually it's from fecal matter that has been dispersed in the air, usually from rodents, so it becomes aerosolized. And I mentioned this term nosocomial. This is referring to diseases that are acquired during a hospital stay, and this can be from anything derived specifically from a surgical procedure, equipment that is not properly sterilized or disinfected, the personnel not properly washing their hands, and also exposure to this whole new wave of antibiotic-resistant microbes. Now, this is a staggering number here, 2 to 4 million cases per year with approximately 100,000 people dying, and almost a third of these could be prevented. And I know these numbers are much higher now. Most of them involve the urinary tract, a lot of UTIs, also cases of pneumonia, especially for old people who are admitted to the hospital. They're already immunocompromised, and they're very vulnerable to developing respiratory infections. Also surgical incisions, so wounds that aren't healing properly. The most common organisms involved are the gram-negative. So these are the ones that seem to be very stubborn, that persist. We can add a few to this list. Mycobacterium. Also, we're seeing Enterococcus as being on this list, and as well C. difficile. So Clostridium difficile seems to be kind of climbing the list as far as a hospital-acquired infection, and it's from the result of antibiotics, so it's an antibiotic-induced colitis, and C. diff is normally present in the gut, but it starts to overgrow when patients are prescribed antibiotics. So 39% of these infections are usually, the source here is from urinary tract or UTIs, also surgical sites, respiratory, some meningitis, GI, skin, and also cases of sepsis. So it's so important, and I know you guys are going to be taking a bunch of classes that will teach you on how to practice these universal precautions, but these are a very stringent set of measures to help prevent the spread of hospital-acquired infections. So this is from patient to patient, from patient to medical healthcare worker, and from also worker and doctor to patient. And this is based on operating on the assumption that all patient specimens could harbor infectious agents, so you have to treat them with the utmost care. Now, there are a set of scientists that we call epidemiologists that will study the frequency and distribution of both infectious and chronic diseases, and they will work to collect, analyze, and report data on specific measurements such as the occurrence, the mortality rates, morbidity, how they're transmitted, and all of this information is required to be reported to the hub of infectious disease, which is the CDC. So Centers for Disease Control in Atlanta, Georgia. You guys can click on the website and check it out. I posted this as a source. I recommended that you guys navigate through here even for your discussion post. So anything that has to do with any infectious disease, type in the keyword and you'll see some great, accurate, valuable information that will come up. Now, the epidemiologist will track prevalence, which is the total number of existing cases with respect to the entire population. Usually it's a percentage. Incidence, the new cases over time, and then mortality and morbidity. Mortality is, just remember, deaths. Morbidity is the people who are affected or afflicted. And as far as the distribution of disease, so if you hear these terms, a pandemic, for example, the bird flu, there's a concern that bird flu could become a pandemic. That's worldwide, so that's international. Epidemic is when it's basically spreading beyond what was expected, sporadic, at irregular intervals, and then endemic would be a particular infectious disease that is always present. So a disease that exhibits a relatively steady frequency over a long period of time, you always find it there. So cases of typhus are endemic in some areas of the world. Rhinovirus, which is cold virus, is always circulating in the population. So there are some infectious diseases that are endemic, and these are just cartoon images displaying that. Pandemic, worldwide. Epidemic, spreading beyond what is expected. Endemic, it's always present along this particular area of the U.S., along this coast. Sporadic, irregular intervals. And then lastly, with regards to the interactions between microbe, pathogen, and host, are these set of postulates proposed by Robert Koch, an early microbiologist. And I would like you guys all to remember this set of postulates, because I can promise you that I will ask you about them either on an exam or in some form. And I would like you guys to also be able to think of exceptions to each of these. So Robert Koch, and this was over 100 years ago, when again, there was this time where no one believed that there was anything smaller than what we could see, right, with the unaided eye. And he developed a set of guidelines to prove and link a particular microbe with a specific infectious disease. So he said that if you thought that there was a particular microbe that caused all cases of a disease, let's say you suspected there was one pathogen that made everyone sick and gave everyone the symptoms associated with tuberculosis. He said that in every case of tuberculosis, that you should be able to take a look at the sputum in their lungs and find a particular microbe. You should be able to find Mycobacterium tuberculosis in every case of TB. He then said you should be able to take the microbe out of sick people and grow it in the lab, cultivate it in the laboratory environment. So take it out of that sick individual, isolate it, inoculate a sterile flask of media or a sterile culture tube, and let it grow. Then he said after you culture it, you should be able to inoculate or infect a healthy host, which we don't do these types of experiments with people. You can do them with rats or mice because no one cares about killing a bunch of them. So let's infect a healthy mouse or a rat with Mycobacterium tuberculosis, and let's see if that animal develops the same disease. If we do see the same disease, then he said lastly, we should be able to, after they show symptoms, they're coughing, then we should be able to re-isolate that pathogen. Now, can you guys think of any exceptions here? Well, I'm going to go through and give you guys examples. So number one, find evidence of a particular microbe in every case of disease. Well, what about pneumonia? So out of 20 people who have pneumonia, you will not find the same microbe in every case. There are bacterial causes of pneumonia. There's streptococcal pneumonia. There's mycoplasmal pneumonia. There's also viral pneumonia. So you're not going to see the same microbe in every case of disease. Also, number two, isolate the microbe from the infected subject and grow it in the lab. Well, Can we grow all microbes in the lab? Not necessarily. There are some that have these special growth requirements. What about a virus? A virus is very difficult to cultivate. You need a specific cell line. You need additional advanced tools to culture a virus. Bacteria are a little easier, right? They don't need, except for rickettsias and chlamydias, they don't need a host cell. They can replicate just in pure culture. What about the third one? Inoculate a susceptible, healthy person and see disease. Well, if you have immunity, you've been previously exposed. Let's say you've been vaccinated. You're immune. You will not develop disease, right? And then the last one, re-isolate the agent from the same subject. Pathogens, as they replicate, will change over time. It may be that the pathogen that we find in the end is slightly genetically different from the one that we initially were working with. So there are changes over time, which we call mutations, that can be beneficial or detrimental to that particular organism or to that pathogen. So remember these exceptions, they are important, and let's give Robert Koch some credit. He devised these a long time ago, so it was a framework and a foundation with which now we have built up exceptions based on current technology and current observations of how pathogens work. So that concludes chapter 13. Again, you guys have an exam this week, so for the hybrid, it is in lab. For the online crew, you have to complete the exam at some point during the week. Please let me know if anyone has any questions, and I will see you guys next week for another lecture.